One thing that most educators want to know is whether their students improve when certain interventions or techniques are implemented. Quasi-experimental research can help answer this question. But first, a few words about the term quasi. Unlike non-experimental studies, quasi-experimental studies involve some degree of control or manipulation of variables by the researcher. However, they lack the strong control and safeguards of true experimental designs. Tentative cause-effect conclusions can be drawn, but other explanations for findings are plausible. Thus, we don't really talk about interventions working or causing improvement in these designs. Instead, we generally say that the interventions are associated with improvement. Although some researchers would place the one-group pre-test post-test design in the category of pre-experimental research, for our purposes we can consider it quasi-experimental, as it does have some of the features of an experiment. In a one-group pre-test post-test study, a group of participants takes a pre-test, receives an intervention, and then takes a post-test. A change in mean scores from pre-test to post-test indicates that the group, on average, changed from beginning to end of the intervention. The intervention might have caused the change. However, other explanations for the change are possible. For example, the group might have matured. Alternatively, other factors occurring at the same time might have influenced the change. To be on the safe side, it is best to say that the group improved when the intervention was implemented, rather than that the intervention led to improvement. In this type of design, determining whether the group changed from pre-test to post-test involves calculating the mean for each time point, pre-test and post-test, and then calculating a statistic that compares these two means. The most common statistic is the student's t paired. This is very similar to the unpaired t-test involved in studies that look at the difference between two group means. However, the paired t-test looks at two means from the same group. If the t statistic is significant, the results obtained in the sample is likely to reflect the result that would be obtained in the population. In other words, the results are unlikely to be due just to chance. Let's talk a bit more about statistical significance. Statistical significance is the likelihood or probability of obtaining a result in the sample if there were no true result in the population. Remember, sampling error is inevitable. Thus, sometimes you will get a sample that behaves or responds differently to a situation than the entire population would respond. Significance testing will give you the chance of your sample being very different from the population. More specifically, significance testing involves testing the likelihood of the null hypothesis being true, that is, there is no difference or change in the population, in light of the data obtained with the sample. Some changes or differences in the sample are too big to be purely due to chance, so we would conclude that these changes or differences are significant, that is, likely to reflect the outcome that we would get with the entire population. Now, note that this is purely hypothetical. The true population result will never be known. Therefore, we must use probabilities to reflect our uncertainty about the truth of things. A low probability, or small p-value, indicates a significant result, something that is unlikely due to sampling error, but instead represents a true, that is, population result. As a convention, researchers use the p-value of less than or equal to 0.05 as their significance cutoff. This represents a less than or equal to 5% chance of saying that the sample result reflects the population result, when in fact there is no result in the population. 5% or less is sufficiently small. We are comfortable saying that the result in our sample is real, even though we can't know for sure. Here is an example. A teacher has a class of 20 second grade students. Over the course of the year, she implements read aloud strategies to improve their reading. She compares their beginning of the year benchmark reading test scores with their end of the year benchmark reading test scores. The class's mean or average beginning of the year score was 50 out of 100. The class's mean or average end of the year score was 80 out of 100. The class improved on average 30 points. What is the likelihood that the same improvement would be seen if the whole population of second grade students received the read aloud strategies and were tested? The t-test compares the size of the difference observed between the means, in this case 30 points, to the size of chance differences that can occur due to sampling error. Remember, even if there were no difference between the pre-test and post-test scores of the entire population, we would expect that some students in the population would improve in their reading ability, even in the absence of the intervention. The t-test will determine whether the 30-point improvement is larger than what we would expect to see just due to sampling error. In this example, the t-test indicates the likelihood that the difference observed was due to sampling a group of second graders that was different from the population of second graders in general. If this likelihood is small, we can conclude that the difference observed is due to a true improvement in the population of second graders from which the sample was drawn. Though remember, this is just hypothetical, as we have not intervened with nor tested all second graders. If we consider the difference to be a real difference, we reject the null hypothesis, that is, no difference in the population, and conclude that the alternative hypothesis, that is, a difference in the population, is probably correct. In this case, the observed difference between means, 30 points, is larger than what we would expect to see by chance with a sample size of 20 students. We reject the null hypothesis and conclude that the improvement from pre-test to post-test in this sample represents a real improvement that would be seen in similar samples from the same population of all second graders. Thus, we would say that the sample of second graders showed a significant improvement in reading. Now remember, just because a result is statistically significant doesn't mean that it has practical significance or meaningfulness. One way to measure meaningfulness is to measure the size of the difference between two means. This is one type of effect size, indicating that it's the size of the effect or outcome that you have obtained. A popular measure of effect size is Cohen's d, which is a standardized measure of difference between means. Here, standardized means that the size of the standard deviations is taken into account, so that the resulting effect size can be compared to effect sizes from other studies with samples that have different standard deviations. The formula for Cohen's d is mean 1 minus mean 2 divided by the pooled standard deviation, which is similar to the weighted average of the two mean standard deviations. Cohen cautiously proposed conventions for interpreting d, such as a small effect being anything between 0.20 and 0.49, a medium effect being anything between 0.50 and 0.79, and a large effect being anything 0.80 or higher. However, note that these are somewhat arbitrary. They simply represent fractions of standard deviations. Very important changes or differences can be small, while large changes or differences do not necessarily indicate importance or practical significance. An additional type of quasi-experimental design is called a non-equivalent control group design. In this design, at least two groups are used, so it is an improvement on the one group pre-test post-test design. The two groups are then randomly assigned to conditions, such as treatment or control. However, the participants themselves are not randomly assigned to groups. The groups are intact. They were already a group before the experiment. For example, two classes of students are intact groups. You could assign each class to a condition, but the two classes might differ in other important ways that could influence the results. Nonetheless, the procedure for the design is the same. Give each group a pretest, expose each group to its assigned condition, for example, intervention or no intervention, and then give each group a post-test. Conduct paired t-tests to see if each group improves significantly or not, and then calculate an effect size for each group's improvement to see which group improved more.